synchrotron facility joined, including Japan, Korea, Shanghai, Taiwan, and then Singapore and Australia synchrotron. Okay, and then our design of this consortium is each synchrotron is coming to join this consortium with a research institute. For example, in my own institute, Academia Sinica, is coupled with our synchrotron. And then so that we need the synchrotron to provide the beamline and then access, but we also need a group of scientists that is able to develop further the technology, the imaging technique, and also to process the data. So this has happened to for example, in Japan, it's a RIKEN and the Spring A Center, and Singapore is National University of Singapore, coupled with their SSLS synchrotron. And then over the years, and then it was the inauguration I showed already many years ago. And then most of the synchrotron directors show up to sign this MOU to launch this project, this program. And we even have a 3 Nobel Prize to show their support in this meeting. Okay, and then this consortium we will focus on the imaging technique. So X-ray technique is the basis. So we're going to use X-ray technique to build this the circuitry or the road maps or these, and then we would make this as a structure which is able to incorporate all the other technique, such as either functional, molecular technique, which has no capability to look through the whole brain, but given them a good registry. For example, many people made the example to explain this. It's like we are mapping like a Google Earth. We're showing you the continent, showing the interstate highway, and so on. And then the detailed information can be annotated by information obtained from the other technique. So this is one of the important development we kept in mind is when we develop this thing, we want to leave room for our database that allow other technique to put in their information. And then what we offer is that if you look at just any high resolution technique like a TEM, you look at one piece of sample, it'd be very difficult for you to know where exactly it's from, and then what is the relation with the larger part of the brain, for example. So this is the idea that we have about the integrated or incorporate other technique. But X-ray being the only technique that we can look at the whole brain, and then with a sufficient resolution to show at least the major connections. Okay. So through the years, since 2020, which is affected actually very much strongly affected by the COVID because it is an international collaboration project, and then the COVID exactly prohibit that. So, but still with a very tough, because I personally, for example, was quarantined 4 times, and each time is like 2 weeks because of the travel. So through this effort that we are able to sort of standardize and calibrate all the participating synchrotron facility and the beamlines, and set up a station in these synchrotrons. Okay. So, and then at 2024 into this year, then we are very happy in that we have actually new members like Thailand synchrotron and then the Sesame in Jordan joined us officially, I think in 2022. And then now in the next month's meeting, Poland synchrotron Polaris, and then the computational research institute called SANO will pair together and then come to the meeting in the south of China to sign the MOU to become an official members. And then we are also in contact with Brazil, which show very strong interest to join this consortium. So in this way, we'll be soon have at least sort of 10 member countries. And then many of them, for example, in China, now we have two, and then the third synchrotron is in discussion. And then in Japan, we also expected with the Spring A synchrotron, and then the new synchrotron, the Tohoku synchrotron, might also join the consortium. Okay. So, and now this consortium, except continues to take the brain image, mapping the brain. That one of the key objective is through this collaboration, we can develop a new imaging technique. Okay. For example, one of the best example is this one we call it X-ray light sheet microscopy. We call it, we give a name, a fancy name as Maxwell. And basically, it was using the very high precision mirror produced by the Spring A synchrotron, and then use it to focus X-ray into one dimension, and then make it in a very thin light sheet. Then an illuminated sample, which has been labeled by the scintillating nanoparticle we developed. These particles are of the size of about 10 nanometers. And then the beam we can focus, the first test we're using 60 nanometer thickness beam, and then scan through the sample. And then those scintillating nanoparticle was only lights up when the X-ray hit it. So then this type of imaging, even with visible light microscopy on top, look at the emissions of these scintillating nanoparticle. It does give you a very, very good Z resolution, which is defined by your light sheets. And then in the XY direction is a typical normal X-ray visible light resolution, but we also borrow the same idea of these super resolution microscopy. And by processing the data, we very recently we achieved actually 7 nanometers of the XY resolution, and then 2 nanometers of the Z resolution. So this is manuscript is finished, is prepared, and so we are in the process of submitting this paper. So that will be the first time you see that we have three-dimensional imaging with resolution all below 10 nanometers in all three directions. So this is one example we show without these super resolution deconvolution. The Z resolution is 65 nanometer. You can see from the image. And then, so this is one of the technique. We believe there'll be a lot of potential being the one sort of complement the full field imaging like the one we are doing for mapping. Okay, and then all these imaging strategy was published. The first publication was in 2017. Okay, so you can check it out. At that time, we outlined how we are going to do this brain mapping. And then more recently, 7 years later or 6 years later, we published another one with much more concrete line of thought, and supported by many preliminary data to show that this is a viable roadmap to the large brain. This large brain actually specifically mean that the human brain. Okay. And then again, then I come back to the result of that is then we're going to have a lot of data. Okay, so this was this picture was taken at the 2016 the SFM meeting, the largest neuroscience meeting, and in the U.S. And then our partner, Professor Anxin Chang, was giving a presidential lectures. And then he outlined that he announces that we are going to use synchrotron X-ray tomography to do this mapping. And then this paper was a report by Nature, and then showing outlining all these things we mentioned. But the title of this is Big Brain, Big Data. So the data, as I said, if we collect it by X-ray with a 0.3 micron resolution, in all three directions, the amount of data we will collect will be 1 exabyte for one human brain. And remember that I will tell you later, so okay. But then this is another example because Professor Anshin Chiang, that was the one giving the talk, he was an expert of the Drosophila brain, and he spent something like almost more than 10 years to look at over millions of 5 millions, I think he said, Drosophila, taken out one by one and using this tissue clearing fluid that he invented to 2002, and then make the Drosophila brain transparent, and then express the floor for GFP, for example, and then image it by confocal microscopy. And it took him more than 10 years to finish only 50% of the neuron. So then he said, just jokingly, by this speed, the Drosophila brain has 130,000 neurons, but human brain has 55 billion. So if it takes him 20 years, then it would take 17 million years for him to finish the human brain. And then he was not doing this by one microscope. He has his lab has at that time 10, and now it's probably 30, 40 confocal microscope and imaging this simultaneously. Okay, for X-ray part, we believe that, as I mentioned to you, we believe we can do it much faster. For example, as I say, 3 years. And then, but the problem is, the most challenging problem is the data. Okay, the single fly brain is already 100 gigabyte, and the mouse brain is hundreds of terabyte. And then we want to map, if we want, because this brain, we are only imaging few percent of the neuron because of the staining method, Golgi stain we use. But if we complete this by the map with, say, 100 brains, then it will become tens of terabyte. And then when we do the human brain, then human brain, the size is already 2,000 times more than the mouse brains, and you calculate. So means, okay, we have to accelerate this, okay? And here I'm showing some examples of that we do, for instance, for the Drosophila brain, and then we are able to stain the neuron without actually sectioning even the skull. So, and you can see by sectioning, we can see inside this is the Drosophila brain and all the things we can trace. So I would speed up because it's the end. So the rest is just showing examples. In the last couple of years, what we have done. This, for example, is a mouse brain. This is the first time we try to put the blood vessel, which is the brown, there's a gold features, with neurons, which is the green color features. And then, of course, these are two different brains, but we're sort of trying to merge it together and to show how exactly they are related, or how roughly they are related. And then this is the projection image of these things, and you can see also the blood vessel, which these are straight lines of the blood vessel which is stained by Golgi, not because of our perfusion. So you can see it's not so well connected. And then so this is how it looks by one completely slice. So by this slice, if we take the whole brain, then we can do by the whole brain without cutting. Okay. And then these are the high resolution image. So I will conclude. So it's almost 1 hour with my talk. And then after this, I will play this movie when I was waiting for your answer. Okay, the progress of this consortium is, after many years, after 4 years since inauguration, we are finally able already to take on the human brain. So we expected the first donation of the brain will arrive hopefully at the end of this year. But right now we do not have the speed to image the whole brain at once. Okay, so we are still waiting for few developments. Once these development installed, we project that we can image the whole brain within a few months. Okay, But before that, we would try to look at a few specific region. For example, if we think we can spend just instead of months, we can just take 1 or 2 weeks of beam time, when we can map out the complete hippocampus region, for example. And that will be actually already quite significant, because that will be the first such a comprehensive mapping of any significant brain regions. Okay, then I would not discuss. I would just show you, besides other than the technology, the imaging, and then the biological data from the brain, and then this connectome map. There are many other possible technology impact. For example, the imaging, as I mentioned, and also the new imaging technique that we developed due to in this process. And then we can do in the future, for example, spectral microscopy coupled in with elemental chemical mapping, and then so on. And then, but most interesting thing is actually the neuromorphic computing. This is not very mature yet, because I think most of people still have no idea how we can learn from the human brain to make a better computer. And the more neuromorphic computing is the one coming closer to mimic how the brain works. Okay, so I just list a few things I gather. For example, the first neuromorphic computer was actually built by IBM, and then the first computer was actually sold to American Air Force. Okay, and I have no idea how it's useful or what it does better than the regular silicon-based computer. It's different than quantum computing. Quantum computing, people actually know quite well what it can do best, and then of course it's not everything. Okay, so and then for example, we just recently sent out a proposal to the Chinese government, and which is pushing for this AI for science project. And then within this project, we're asking a group of computer scientists to help us to develop the necessary algorithm or computation tool to handle our data. The first task, for example, when trying to do a sparse projection reconstruction, means that instead of using filter back projection, which require hundreds, if not thousands, of projection to do the reconstruction, and then hopefully by machine learning, we can do with much fewer projection, which is not only increase the imaging taking time. For example, if we can do, which we have already demonstrated, that this reconstruction can be done with 20 or 30 projection, and our goal is push it to under 10 projection. And then compared to 1,000 projection, the acquisition time is literally 2 orders of magnitude faster. Okay, so that if you just calculate this way, that means instead of taking 80 days to finish a human brain, then it could possible that we can do it within few days. Okay, and then how do we extract the wire diagram, which is the information we need? And from these exabyte of image data, then what can we extract these connection information? Okay. All right, so I think I will stop here, but by showing a few images or movies when I go to the very end. Okay. So here I will conclude, because we modified that. So because this SYNAPSE project is really restarted after the SYNAPSE after COVID pandemic, but within the last few years, within the years during the COVID, instead of pushing for the large scale mapping, we focus on develop the technology. So now we are very confident now when we started with all the investment on the hardware and the computation, we'll be able to finish the first brain mapping before 2026. Okay, in 3 years. And then I will have this sort of a joking slide. It's okay. There's an old saying that go fast, or if you want to go fast, go alone. If you want to go far with friends. Here we want to go fast in terms of it means the imaging, and want to go very far to reach the human brain. And then we are gonna go with all these friends that have been collaborating with me and helping this project. Thank you very much.
Oh, thank you. That's a very fascinating talk. Are there any questions for the speaker?
Okay. Oh, sorry. Okay. Yes.
You can either unmute yourself and ask or do the raise your hand thing, like I
Okay.
just did there.
Okay. Yes.
Uh, well, let me ask a few questions if I can.
Yes. Mhm.
Is it possible to get the resolution at a wavelength where you could probably get some chemical signatures of the neuron on a spatial basis? So is like doing this in the IR.
Yeah.
Does the wavelength mean that you can't get the spatial resolution? Would that be a deal breaker there?
Actually we have of course these kind of idea they always come up and then within some discussion. But to be honest, that we have actually no idea how we can implement that. For example, and then being an experimentalist, and then dealing with a large facility, then actually we have been all educated to reduce your ambition to these things.
Yeah.
Okay.
Uh,
So for example, that's why I said the SYNAPSE was proposed with a very in terms of really front end technology is very conservative because all this parameter I show you is very conservative. So, but that doesn't prevent us, for example, all these consortium, because right now, for example, we are already setting up a few beamlines because we have developed the technique during the last few years. So once it's implemented, it could be so fast. So we actually do not require that to separate the brain, one human brain, as we proposed the very beginning of Synapse, to different facility, and then everyone collected the data, and then finally we put the data together into one place and do the process, and then make a database of these wire diagrams so that can be shared by everyone else. So because of the COVID, that we are forced to do the work at home, so we decided, okay, we will try to increase the performance instead of doing the mapping. Okay. So the advantage of that is that get us to learn another approach, strategy is when we are talking about all these facility, then I ask all these scientists because they all understand once they join this project, very soon they will be doing extremely boring job because most of the job is actually sort of can be replaced by robot or by AI. So what I encourage them instead is let's try to develop your own idea. How can we improve the speed? How can we add a new information such as the one you proposed? So can we use X-ray or in a different? I was even thinking about can we use the modulation of the wavelengths for something like that. For example, because you want X-ray to be penetrating, can you modulate the wavelength to give you something to excite, for example, the vibration spectroscopy. So these are the things. So I think we just request asking for our partners to work on this because in the future, I think once we finish the first brain, because this first brain is likely to be uh, completed in China because it'll be probably a Chinese donation. The donor is probably Chinese, and then the China or almost every government does not allow you to transport the human tissue to other country. So if we have that, we most likely will be doing with a few synchrotron in China. But then at the same time, we can use this because to complete a map, we need hundreds of a frame. Okay. of course, it's not like you have to wait for 100 brain to complete a map before it's useful. In every step, even if we image a very small region, because these are the information never being seen, known to be in medical or neuroscience community. So they will be, without a question, will be very useful. So this will allow us to say, okay, each synchrotron, for example, if the African synchrotron is ready, then you can acquire your own brain, and then with one beamline, and then you can do probably 10, 20 brains a year, and in automatic fashion. And this data will be collected in the future, being a commitment to be this consortium to put together, and then this final database we promise will be shared by the world. Okay. So in this, but at the same time, that will allow each synchrotron to look at, for example, any specific disease that is interest most to your country, for example. You have a high population with this specific disease, and then you can image at the same time the healthier brain, which is donated due to private car accidents or whatever. And at the same time, you can also collect information from this diseased brain. Okay, and then slowly we can put together a very complete map. And then at the same time, that you can also encourage your local medical community or something to use this not completed yet connectome map and to do their research to get new findings. Okay, so that's my very long answer to your short questions.
Okay.
Yes.
Any other questions? Trevor, did you have a question? I see you unmuted yourself.
No, I didn't really have a question, but I thought it was an extremely interesting talk. And is the camera that you were using, the advanced cameras that you're using, they're all in China, are they?
No, the Chinese synchrotron, Beijing synchrotron has a project and trying to make. It's not actually advanced. For me, I was joking to these are good friends. I was joking. No, they're just making a bigger Russell number pixel. So the next version they are going to be rolled out is probably 6K by 6K. But I told them, if you leave it to the industry, the commercial industry, it'd be much faster, which is the approach the Spring A, for example, took. I'm actually now talking to you from Spring A. I was in the beam time experiment with my team, and then this is the last day of our beam time. So, and then I knew that they actually collaborate with Sony to make their detector for free electron laser, and then also for this. And then I think that's also a very effective approach. But I think China does this for a specific reason, because they do not want to leave a stone unturned. They want to make sure they have all the possible technology to prepare for a scientific sanctioned war. So I think this camera, from my own understanding, it's really nothing really advanced of they have developed this high pixel number camera for the photon counting device, but that's not really necessary for our X-ray imaging. That's more for like a diffraction imaging or something like that. So I think the camera is not really an issue. I think even without waiting for too long, we will find a camera with 10K by 10K pixel numbers. The problem is it'd be more difficult to make it. They call it scientific grade. But again, I do not believe that is very important or essential. Okay. So anyone with a probably reasonable technique or something, you can buy the commercial chip and try to make a CCD yourself. That's within not only that doesn't even require a consortium work, but probably any facility, and then you can find that 2, 3 scientists can already do this job. All right.
Thank you.
Thank you.
Leo, did you have a question? I see that you have
Yes, Lawrence, more than question is a comment, if you allow me. First of all, let me introduce me to the speaker. I am one of the people behind a project of a new synchrotron in the Caribbean region.
Yeah, I was told by. Yeah.
Okay, so and we are in the strict contact with the colleagues of the African Light Source. Now, your talk was very interesting and stimulating, but of course, to some extent, made me think, well, this guy is presenting us dreams which for our regions are out of scope. But instead, I would say, I would like to comment how one can make this project also interesting for regions where we are not in condition of participating in an active way to your research. You mentioned a certain interaction with medical community. And medical community often in Africa and in Caribbean is strong. We are thinking to have a synchrotron built in 5 years, 10 years from now. But for example, there is all a work of computing which can be very useful for this type of research. So my question is, would not you think that a project as large as your project cannot have also a component of dissemination among the young people to try to build an expertise of few people who can be the core from which in 10 years from now this type of research is feasible in Africa and in the greater Caribbean?
Yeah, thank you very much for the question. And to be honest, okay, I think we would like very much to think along your way, but indeed it's not what I originally planned. Okay, because my less ambitious plan is actually try to call up these few synchrotrons, which are many good friends that I get acquainted or develop a very good friendship over my career. So I said, if I can convince them, say this is something worthy to do, and then everyone just give a certain percentage of their energy, then we can do this job together. But then over these years, with more and more people, and then yes, indeed, we will have to go to this direction, as you mentioned, because that's why I was actually our board. We have this Synapse. We have a executive board, which is, in a way, it's the operating body of this. Which consists of each country will have one membership, and then in this board, and then we can discuss and decide things together. So we decided very early on that, first of all, we will not discriminate against any facility. So when we do this, when you have already a synchrotron, for example, like the Singapore synchrotron, which is not at least in the same performance level as the rest, like the Spring 8. But we decided it's more important to have the spirit of the large synchrotron community and the spirit of working together. And then so that's why my title is, cross the border or borderless approach. So we do not want, because we do not like to see what the scientific border now is actually constructed. So this is also the same idea of Professor Moving Hwu. So he wants to make a even larger consortium just for the brain mapping. So that will include all the different techniques. And he has the intention to make this into a consortium like, if you know, CERN. He said that we will make all the facility open to each other. and then collected data, and then the data is also open to everyone. Okay. So I think this is, so then we have to then come to think about, for example, that's how we practice, and it's quite successful. For instance, after the first membership, then we say, okay, we approached Jordan. First of all, it's like your case here, is we ask you if you're interested. Okay, if you're interested, and then we will talk. Okay, if you're interested, you think, okay, then we'll try to find something that the community, if the synchrotron is not even built, that can contribute to this grand challenge. Okay, so that's our commitment, and then we promise that it will be happen. So if once you show, and then I think the whole board will work as one community, then we'll try to help in any way. First of all, if it's okay to increase the interaction between the medical community and the synchrotron facility, which is also very important, not just in the African synchrotron or Caribbean synchrotron, but every synchrotron. But that is exactly what we did. And then our experience can be translated, if not completed, borrowed, can be translated with the help of the local scientists. So I think this is the good thing is that we have a big enough objectives. This objective is so big, so I think can be shared by everyone, and then it cannot be achieved by a single facility or even single country. So you don't need to feel, shy about, for example, Thailand synchrotron. Of course, this is the first generation synchrotron. The performance is reasonable. We can still appreciate the access. But on the other hand, they are building a new synchrotron, which is on the 2030, if they expect to finish at 2030. So, it's if you are finished in 2030, then it has no use for us. No, the idea, taking the data is one thing. Taking the one human brain data can probably be with our speed that we recently demonstrated, can probably be done by one single beamline at the time of like 2 months. So really to collect this one set of data, it doesn't require anymore all the synchrotron. But we want the synchrotron to work together to see, okay, how can we then move to the next step? That we want to collect 100 brain or 200 brain data. So in the future, it will be like donating the cadaver. For instance, China is already in the process that they have already 3 big brain banks. So asking for the patient to donate their brain is become, without any legal obstacle. So just like you donate your organ for transplant, now they ask you also, do you want to donate your brain? And they have the largest hospital in Beijing actually have this brain bank. They are very efficient, and then they are taking care of all the process. So I think the essence of this is actually try to find a new way to do the image, putting new information, more information. But the most important thing I would like to have this community to work together is actually find a new application. Frankly, even if we constructed the first connectome map, the map from one human, we have, frankly, no idea how it can be used. So this requires our interaction with the medical or biology community as early as possible, so that they can start growing with us and then find out if we have this map, what they can do to their research or to advance the medicine. All right. I hope does that answer?
Yes, but I would like, if you allow me, to make a final short comment, very short.
Yeah.
Because, of course, our project is a project for the future. But I noticed that among the possible collaborators of yours is Sirius. And we are in strict contact with Sirius which is supporting our project first.
Mhm.
And second, I've seen among the authors of the paper on the physics reports you mentioned,
Yeah.
the last one, because we have so many, my eye
Yeah.
fell on the last one. Margaritondo is one
Yeah.
of the most active person in our group. So that I
Yes. Oh,
think that a possibility of establishing the discussion to see what can be done in this direction, small steps perhaps could be considered.
sure. Okay. Yeah. Professor Margaritondo actually is the chairman of our executive board. Okay. And then we take him as actually because at that time we want to have this executive board with someone so like that it's possible to adjudicate it in case of, you know. So we ask Margaritondo Giorgio as the chairman, which does not represent any member country because he's now in Switzerland. It does so. And then if you're talking about anything you want to be, and then I am now assigned to the job of CEO is exactly the one to talk. So if you want me to help you to organize something, and then I will be sort of at your service. Okay. And in fact, I'm going to do a early retire from my job in Taiwan, and then I will be serving this consortium full time.
Thank you.
Yeah, just okay. Draw me a note if you think there's any idea. I think it's not just. I'll be happy to do that. This is what my duty of being assigned by the SYNAPSE
If you can write your email in the chat, I'll be happy to take note.
I'll make sure you get it, Leo.
Okay.
Let me ask just a quick question, hopefully, maybe.
Yes.
I didn't notice the Shanghai soft free electron laser as one of your consortium members. Does the high intensity of a soft X-ray XFEL lend towards the greater speed at data collection, do you think? Or is that
no.
too bright
No,
fry the
no, actually, we have no idea. We have not thought about. Actually, I just before the talk, I actually removed few introduction of the free electron laser because I cannot see in the near future that we will make use of that. Even though this laser is next door, and then we know almost everyone that direct, but they did as a facility they did not participate the SYNAPSE yet. So this will be mainly the synchrotron work, at least for the first phase. As after we build the road map, okay, of course, the free electron laser could probably in the future give us for instance, molecular sensitivity to identify whatever protein is and where and so on. But that is not what we concerning right now.
Gotcha. Okay. And then I guess one comment. I mean, you do one brain, but there's got to be some brain heterogeneity in a global population. So you probably have to be doing a whole bunch of brains before you really learn anything biologically interesting.
Yes, exactly.
So
yeah.
Well, if there's no further questions. Go ahead. I'm sorry. Go ahead.
So that's why we are not. That's what I said. Okay, we said the first one brain is just sort of like symbolically, said it really to make it's much bigger than I make the one brain already sounds very challenging because by the data side, but then think about then to have this reasonable map. At least we need something 100 because of the Golgi stain 3% to 5% at most of the neuron. So you see, okay, even if they are completely random to build a map, how many do you need? Even in a statistical sense, you probably, an order of 100 of them. And then there's no way one facility can do. Okay. And then that's good. Actually, then that allows, for example, that you can look at the African the local populations and then something like that, then the data will be much more diversified and then will be much more interesting. Yes.
Yeah. And then so I guess at each light there's a sort of a standard protocol for collecting the images and the standard workflow and protocol.
Yeah,
So that's
it's
something
not,
that we probably need to talk about in terms of
yes,
training.
yes, yes. And then of course we hope, because at the beginning we are inherited this few beamlines. That's what the work our group did in the last few years, that we went there, and then with their help to implement the hardware, and then implement the software, so that we can take the data with a very similar quality. Because original idea is we want to have a one brain taken into several pieces, right? So when get to that, then we have to make sure the data quality is good enough. For example, that you have to maintain the same resolution so that we can put the data together, you
Yeah.
know. So and then but then we compromises. Okay, we calibrate, for example, this beamline, because okay, like Singapore is lower energy, so in order to get to this, we need to use a wide beam and so on, and then the speed will be very different. Okay,
Yeah.
different beamline. So this way we can calculate, say, okay, then maybe we don't give you such a big piece because then it will be behind. Then we'll give you a smaller piece. But our agreement is we don't, discriminate people, but how much we're gonna work together. Then we don't tell, okay, oh, your facility only contribute 2%. So in the credit line, we will say, okay, no, we're not gonna do that. Okay.
Right. So this
Yes.
is a little bit to Leo's point. This is actually a situation where, well, every little bit helps, and it
Yeah.
actually
And then
it's almost an advantage that only a few facilities will do the work, because you need a very tight quality control on the methodology.
yes.
That makes if
But,
that I mean
yeah,
maybe that makes sense, maybe that doesn't.
but that's reasonably technical, because I was helping Shanghai Synchrotron to build a program to install the brain mapping in the synchrotron.
Yeah.
So in this proposal, I already tell them, okay, I want you to take the responsibility, because with the funding, to support the newcomers. For example, for those synchrotron like yours, you're building, and then many synchrotron during their construction phase, one of the typical practices, they send the scientists outside to work. So I said, okay, if you agree to do this, then you should think seriously, allocate resources to accommodate this visitors or young scientists coming to work with you, not necessarily to learn, but also to contribute.
Yeah.
Because when they come in, then there be valuable manpower, and then they can acquire the experience, and then probably even before the synchrotron is built, they can already engage with their local scientific community to think about how they can use this data.
Yeah. Yeah.
Yeah, that's definitely we have the plan, and we're totally willing to do this.
Okay.
Yeah.
All right. Well, if there's no further questions for the speaker,
Yes.
I will conclude the session by just saying thank you,
Yeah, you're
and
welcome.
that I'll continue to be in touch with you, because I've
Yes.
written down a bunch of ideas
Yes.
that
Yes. Great.
both in terms of working together and then my own sort of scientific curiosities.
Yes.
I'm curious about this XFEL
Yeah.
business,
Oh,
because
okay. Yes.
I don't know if it's sort of too intense to sort of you destroy before you image, like in protein crystallography to say
Yeah, yes,
destroy
this,
before diffract before
yes,
destroy. So,
yes. But yes, we actually I did work on free electron laser for on and off for several years with in the spring A. We have actually published a few things. So I know exactly what its limitation and what its capability. Yeah, so
okay.
just okay, so you're welcome to discuss for idea. We can even ask for example, the spring A scientists to collaborate.
Sure.
Okay, so this is the idea. So we don't we just want people to participate. We do not really need to discriminate whether you can bring in anything. Okay.
Absolutely.
Yes.
Okay.
Great. All right.
Well, thank you for your time.
Right.
And thank you everyone for attending.
Yeah. Thank you very much for being in the talk. All right.
Okay.
Okay.
All right.
For me, it's good night.
Okay.
So
yeah. Good night. And for
Yeah,
everyone
thank
else,
you.
good evening. And for myself,
All
I guess
right.
it'll be good afternoon.
Okay.
Talk to everyone
Ciao.
later. Thank you.
All right. Bye.
Bye.